Hi there, my name is Dr. Stewart, and today we're going to continue talking about viruses, and we're really going to focus on the two different ways in which viruses can replicate. And we're going to use these bacteriophages as an example. So as a quick review, if we look down here, here's an example of a T4 bacteriophage. The bacteriophage is a virus that infects bacteria. In this case, this bacteriophage T4 is going to infect E. coli, that's its host. These guys have a really complex shape. Here is the capsid up here. The name of this little stalk, I forgot to mention this in the previous lecture, that's called the tail. So this is the tail right there. And these little leg-looking structures, those are called tail threads. And they are located right there. All right, and so we're going to focus, now this is a naked virus, right? Remember the two different types of viruses. You have the naked viruses, which just consist of the capsid, which encloses the viral genome along with some enzymes. And then you have enveloped viruses, which consist of the same capsid and the genome with enzymes, but it also is covered by a layer of cell membrane, which it stole from its host, which is really messed up. Okay, but we're going to focus on the replication that occurs by these naked and complex shaped bacteriophages. Now, the first way in which these viruses can replicate is called lytic replication. So L-Y-T-I-C, lytic. All right, and this replication occurs in a couple of really specific steps. The first step that will happen is called attachment. So we'll draw this right up here. Attachment. And attachment occurs when, we've kind of already talked about how this happens, when this bacteriophage is passively floating around. Remember, these guys can't swim actively or move around. They're just floating around in the environment. It happens when this T4 bacteriophage happens to come into contact with the cell wall of this E. coli. This T4 bacteriophage is going to be perfectly matched to the cell wall of E. coli, and that causes it to attach onto it. It's not going to attach onto any other type of cell because it's not matched for that type of cell. So if it comes into contact with our cells, not a problem whatsoever. So the first thing that will happen is that T4 bacteriophage, which I've drawn as this little green structure right up top, is going to come into contact, and it's going to attach onto the cell wall of this E. coli, and it's going to do this by these tail threads, which hold it on into place, and the bottom of the tail is going to attach closely onto the cell wall of E. coli. So here we're going to have the little virus attached right on, okay? And inside this E. coli, this is worth mentioning now, E. coli is going to consist of its own genetic material, which it's using to, do the things that E. coli likes to do. It uses this genetic material to make proteins, right? And it's going to replicate that genetic material each time it divides, okay? So inside the E. coli bacteria, you'll have this blue genetic material. So we're going to call this bacterial genome in blue, and that's going to be located right there, okay? Well, after attachment, the next thing that needs to happen is that the virus needs to insert its own viral DNA into the cell of its host, so in this case, the bacteria. That process is called entry. So after attachment, we're going to have the next step of the lytic cycle, and that's called entry. And you're going to have the same E. coli bacterium located here. Okay, we're going to have the same little T4 bacteriophage right there. But now the virus gets its genetic material inside the E. coli bacteria. It does this in a really kind of nifty and scary way. If we look down here, it's a zoomed in version. What will happen is that this T4 bacteriophage releases a very specific enzyme called lysozyme. And lysozyme breaks down the peptidoglycan, we'll talk about that later, in bacteria. It breaks down the special tough cell wall of the E. coli. And then the tail of the T4 bacteriophage is going to pierce into the cell like a hypodermic needle, and the genome of the virus is going to enter into the bacteria. So it's really like this nasty little mosquito bite, like a needle goes into the bacteria, and that causes the viral genome, which I'm going to draw in black, to kind of enter into the E. coli bacteria. Another thing that comes in here are these viral enzymes, and you'll notice that I'm not drawing the blue bacterial genome anymore. That's because these viral enzymes literally cause that viral genome to disintegrate. So I'm just going to draw a little bit of that genome because it's literally killing all of the genetic code that exists in that bacteria. That's really messed up. Without the bacteria having its own genetic code, it can't make any proteins. It can't do the work that it's supposed to do as a bacterium. So the virus has really just hijacked the cell altogether. And this process is called entry. Well, after that occurs, you've got this viral genome inside the bacteria. With these enzymes, the bacterial genome is now destroyed. Well, the next thing that happens is called replication. And so synthesis. And so with synthesis, what happens is that the ribosomes that exist inside this bacteria, these are the bacterial ribosomes. Now remember, ribosomes, their job is to make proteins using the genetic code as a guide. There's no more bacterial DNA or genetic code in there. So the ribosomes aren't going to make bacterial proteins. What they are going to make is viral proteins using the virus's genetic code as a guide. So now we start to make all these little pieces and bits and parts of new viruses, right? And that is called synthesis. And so these ribosomes are using the viral genetic code as a guide, and they're starting to make all these little pieces of new viral particles. We'll get some tails here, some new capsids, more tails over here, capsids right here, maybe some tail threads. This bacteria is now just filling up with these new pieces of viruses. Now the next stage, after all these pieces of the viruses are produced, is those pieces of viruses start to assemble into new T4 bacteriophages. That part of the process is called assembly. So here you'll have the same little E. coli bacteria, but now inside of it you have all these newly formed and assembled T4 bacteriophages. And I'll draw them in all kinds of different orientations. You'll have the capsid, right? You'll have the tail threads. And inside each T4 bacteriophage, you're going to have a new set of viral DNA. Okay. Now, after there's a couple hundred of these that have been assembled, the enzymes that are still existing inside this E. coli bacteria are going to cause the cell wall of the bacteria to lyse or rupture. So really this bacterium just explodes in a process called lysis. Right, so it's just exploding. And as it explodes, it's going to release all these newly formed T4 bacteriophages into the environment, and we just completed the life cycle. All these new T4 bacteriophages are then going to float around the organism until they happen to attach onto a new E. coli bacteria. I guarantee you there's a bunch around because you're never going to find just one E. coli bacterium by himself. And then once they attach, the whole process starts over. And that is your lytic replication cycle. Attachment, entry, synthesis, assembly. This whole process takes like 20 minutes. That's crazy. Think about it. 20 minutes for hundreds of new viral particles or virions to be produced, explode that cell, they're going to infect hundreds more. So it's quite a rapid process. Now, Another type of bacterial reproduction or reproduction that can occur is called replication. It's called lysogenic replication. We're going to use another bacteriophage as an example, one that also infects E. coli. This is called a Lambda Bacteriophage. So this is Lambda Bacteriophage. It looks a lot like your T4 bacteriophage. It just doesn't have these little weird tail threads. It just has the capsid attached to a tail. So you're going to have this capsid, which contains the genetic code attached to the tail. First thing that will happen is you're going to have the attachment of that Lambda bacteriophage onto the E. coli bacteria. Same process for the T4 bacteriophage. It just matches up perfectly with the cell membrane. Once it attaches, it's going to go through an entry process where it inserts the genetic code of the Lambda bacteriophage into the cell of the bacteria. The viral genome goes into the cell of the bacteria. But here's where things get a little bit different. I'm going to redraw the E. coli's genetic code in here in blue, just like before. Instead of this Lambda bacteriophage destroying the genetic code of the bacteria like it did in the lytic replication, what will happen is that the viral genetic code is going to be inserted into the E. coli, and those two genomes are going to combine. And that's pretty crazy because now the virus is literally genetically coded into the genetic code of this bacteria. And that's all that happens. Now the virus undergoes more of a inactive period where it's not active. It's not going to destroy the genetic code. It's not going to make viral proteins. It has just inserted its DNA, which is now a part of the genetic code of this E. coli bacterium. So there's the blue bacterial DNA, and combined inside of it in black, that's the viral DNA. Now this is crazy. As this E. coli reproduces, it'll reproduce through fission, where one E. coli bacterium splits to form two daughter cells, or two E. coli bacteria. And as that happens, the viral component of the genome is replicated as well. So you're going to have the original bacterial genetic code, and then incorporated to that, you'll have the viral genetic code, which is reproduced. And what's crazy about this is that this can change the behavior of this bacterium. In fact, some really nasty strains of diarrhea, which are caused by E. coli, are the cause or exist because of the viral kind of genetic code that was added onto the E. coli. Before it was infected by this virus, E. coli wasn't that nasty at producing diarrhea in some cases. But some strains of E. coli, which have been infected by certain bacteriophages, this new viral genetic code makes that E. coli much more pathogenic. So this process continues. These cells continue to divide, and that produces more and more daughter cells, which have this viral portion of the DNA. Let me kind of draw this out. All right, and it continues to divide, continues to divide, continues to divide. So we're producing more and more E. coli bacteria with this modified genetic code, but still the viruses are inactive. However, in a process called induction, the viral DNA can become active. This could be triggered by ultraviolet light, it can be triggered by environmental factors, but some trigger is going to cause that piece of the viral DNA to become active. And when it does, the exact same thing that happened in lytic replication is going to occur. That the viral genome is going to command the ribosomes to start producing viral particles. So really, through the process of induction, it's going to kickstart the lytic replication process. So we're just going to go right over here. So through this process called induction, the lytic replication process is kickstarted. The viral particles take over. The E. coli cell lyses, and it releases hundreds of new viruses into the environment. Right? So those are the two basic types of viral replication. Next time we're going to talk about how animal viruses are able to attach and enter on to animal cells, because it's a little bit different because a lot of those animal viruses contain an envelope, which we haven't really talked about yet. Okay? Thank you.